Good morning, everyone. Welcome to our webinar on how to evaluate preclinical and clinical DILI. My name is Karen Dowell, and I'm Marketing Director at InSphero. And before we get started today, I want to invite you to take a look at the chat window on the right. You can use it to ask questions at any time during our presentation today, and we'll address as many questions as we can after Armin's talk as time permits. We also have some polls that you can complete while we wait a minute for everyone to log in. We're going to give it just another few moments here. Okay, I'm going to go ahead and get started. It is my great pleasure to introduce today's speaker, Professor Armin Wolf. Armin is an accomplished pharma R&D executive and board-certified toxicologist. He has more than 30 years experience in leadership roles at companies like Janssen and Novartis. He's currently Chief Scientific Officer at InSphero, and he maintains a dual appointment as a professor of toxicology at the Technical University of Causality in Germany. Armin joined InSphero in 2019 with a vision to help pharma companies take full advantage of 3D human tissue platforms and in vitro technologies in their quest to find safe, effective cures and therapeutics. Today, he's going to share his strategy for addressing drug development challenges and improving translation from in vitro platforms to patients. Armin, I'm going to give you the floor now.
Thank you very much, Karen. It's a pleasure for me to talk about how to evaluate preclinical and clinical DILI. This is a question you hear very frequent in pharmaceutical industry when there's a case and it has to be investigated and to be mitigated. And today I will talk about strategies. I will give you an overview on strategies and I hope to give you some examples how for this strategy and how the testing strategy and how to apply 3D spheroidal microtissues. And particular, I would like to emphasize the impact of this investigation for the drug development process. Let's start by the discovery process and the development workflow. This is really the rocky road to success. It can take 10 years depending on what you're developing, and it costs between 2 and 3 billion US dollar. So it normally starts by a huge number of compounds which have to be tested for drug efficacy. First, you're starting by approximately 10,000 compounds you would need to come down to one approved drug. That in the drug discovery phase, all the classical drug discovery has to be done, the target identification, validation, hits to lead until lead optimization. And then slowly the toxicology starts in this early discovery phases by using predictive in vitro models, selecting candidates which are not in favor, having a bad safety profile. And then when it's narrowing down to a lower number of compounds, then the in vivo toxicology starts, and that's in fact the whole process to come down to one approved drug. Interestingly, it's very often the liver which causes here trouble and which is a major cause for the attrition. We have here the so-called DILI, which is drug-induced liver injury. And DILI is in fact what you observed when you take out the livers of the treated animals, and you look on the histopathology, and there are some very specific pathologies described for the liver, which is the cholestasis, the disturbed bile acid homeostasis. The steatosis is the accumulation of fat and the fat metabolism. Then replacing the number of hepatocytes by a fibroblast type of cells, and it causing the fibrosis. The hepatitis having inflammatory cells in the tissues, and then leading to necrosis and apoptosis. So this is what when I'm talking about DILI, how DILI look like. That's the DILI phenotype. So we know about that. We know that about between 35 and even more percentage of all compounds which will be tested in preclinical animals, that they will have a DILI case, and there's a dropout depending on the company where you are, on your underlying chemistry. It can be lower, it can also be higher. So it's depending really on the pharmaceutical company. So during the preclinical phase, you're eliminating already compounds based on DILI. And once a compound's passed this preclinical phase, you may lose your compound then in the clinical phases from 1 to 3. And this is in fact the situation. And when a compound is passing those phases safe, then it can happen, and it happens frequently, that the compound is lost due to some idiosyncratic DILI, which means it's not predictive. It's an unpredictive type of DILI. By all the means, you have to test DILI, and then some companies, some drugs, they have to be withdrawn from the market. So interestingly, that from the 90% of the compounds who are tested first in human, they have an issue then in the clinics, and they are lost, and this is simply to the lack of efficacy, but also safety has here a huge potential safety issue. Which means there's a tremendous need for improved translation to man, and also to improve the 2D models which are used, and in terms of in vitro models, the 3D spheroidal model turned out to be here superior and have some outstanding features. I would like also to reference Thomas Hartung from the Johns Hopkins University at the ACT last year. He presented a nice picture, and I had to take that picture because it's comparing fried eggs sunny side up with 2D cell cultures, where the morphology of the cell is completely changed. Once you put a primary cell on a dish, the morphology is changed, it's flattening, there's no interaction between the cells. And I think this picture reflects very well this property of the 2D cultures. So 3D are superior in this respect, and I would like now to explain why I believe that 3D could be superior in terms of their physiological function. And I would describe spheroids, or microtissues, as they're also called, as the smallest functional unit of the liver. It has all the cells which are also in the liver. You can compose the microtissues depending on your purpose, with liver endothelial cells, with Kupffer cells, or hepatic stellate cells. There is some communication, there is some interaction under these spheroidal conditions, which is not necessarily the case when you do that in 2D cultures. It maintains the basic physiological function, which are drug metabolism. That's the task of a liver. There's some transport function, uptake, secretion. There are some synthesis functions, bile acids, protein, and so on, and secretion. So these are the functions which are maintained, which the model has been characterized for and maintained. You simply look on the proteomic profile of a native liver and a 3D culture. You see that in the upper right panel, that there is no difference in the heat map of these for these two conditions. But when you compare that to DILI culture, there you see an absolutely different heat map, and which is inverse, in fact, to that what you see on the left side. So this is a specific property of the 3D culture. It comes from the 3D architecture, and this comes from the self-assembling of the tissue. As I said, there is no artificial matrix of these during the production process. There's cell-cell adhesion from the beginning on, and these cells, they're secreting their own extracellular matrix. So this makes the model unique. Another feature of the unique property of the model is that the model has a long lifespan. It can be maintained for 24 days and longer. And I think the longevity is an important property also for the drug discovery evaluations, because ideally one is when you're testing toxicity, you're testing that exactly as you would treat a patient or you would treat an animal. So by daily exposure, having a peak trough situation and so on. So that all can be done under such a model which has a very high longevity. So the model is also translatable to other species. So there's a rat, dog, monkey model and a human model in the end, where the cross-species effects can be compared, and that's a frequent question of toxicology to understand why the importance of for the therapeutic window, which has to be determined under for the toxicological studies. The model is extremely, let me say, economic in a way. So we have the possibility to look here on one micro tissue in a volume of 70 microliter, and this gives really the opportunity to look to do a lot of study with the cells from one liver. It gives also the possibility to look here on a whole range of biomarker and endpoint which we can look and which are important to look at as a toxicologist. And the model is extremely robust, scalable, has a high reproducibility of the data, and that gives a whole lot of quality coverage of those aspects when we're looking on this model. So 96 well played, we are even higher. We can have also to look on 384 well played formats, so which increases the possibilities of this model. So these are the biomarker. On the left panel, you see those biomarker or these mechanisms which are leading to DILI and which can be investigated. It's reactive drug metabolism, the reactive oxygen formation, inflammation, disease factors, bile acid homeostasis, mitotox, and so on. And on the right side, you see the methods which are used for that purpose, coming from simple histopathology after treatment with a drug to specific biomarker in the supernatants, and doing here some biochemical investigation, and also having here the possibility to look on omics like transcriptomics, proteomics, and so on. So this is a whole repertoire which can be applied for the microtissue. So now let me start with the first application, the DILI hazard identification. DILI hazard identification is a typical approach in an early discovery phase, where the compounds are screened for efficacy, and the DILI screening can be added on top of that. It is a specific type of science. It's data science driven, and the algorithm for these DILI predictions, they based on correlations and on statistics. They are evaluated and assessed based on high specificity and sensitivity. And normally such a screening assay, it has an easy endpoint to follow up. In this case, it's the ATP-based endpoint, and it will not provide some mechanistic information, but it has been demonstrated that such an assay has a high value for the drug discovery, and that was described and published in the paper of Proctor and Williams, and they demonstrated that from the 108 clinically annotated different DILI-inducing drugs with a different potential, they could create the correlation to the respective level of cellular ATP. So that makes sense, since ATP is also an endpoint for which is involved in the necrosis. So we can create here the link. And in this study, the correlation and the comparison to the 2D primary culture was investigated. And you see here on the red circles that compounds, for instance, like bosentan, that they have a very steep curve, dose response curve in the 2D culture, but it's getting very let me say very flat curve in the 2D culture, but very steep in the 3D culture. And you see that also for other compounds like troclitazone and for instance simvastatin. So that points out the value of the model, that it has a higher sensitivity. That was demonstrated that the sensitivity, which was relatively low, 20%, and that could be increased up to the twofold of the sensitivity score, and by maintaining the high specificity of the 3D cultures and compared to the 2D culture, which is the same. So I would like to come to my next point, just moving from the ATP and hazard identification to the risk assessment. That's the next challenge, and here you have to not to look only on ATP consumption. You have to get in some more mechanistic information. This is compared to the compound statistical approach. This is more an evidence-based science approach. You have to look on the individual cases. It will be the process for mechanistic investigation will start by the preclinical data. It will look on the sequence of events that can be then used for the creation of specific hypotheses. And further, these hypotheses, they have to be validated in order to make here some conclusions, and also from the risk assessment point of view. So that will allow here to make to fill the gaps between the in vitro and in vivo when you're coming up with more mechanistic information, which is more relevant to the in vivo situation, and based on those observation, you creating here some correlation between the in vivo and the in vitro data. So one tool which is a very helpful one to better understand the mechanisms of DILI, but also for the investigation of DILI mechanism, it's a so-called adverse outcome pathway. It looks simply on the sequence of events, and it's based on existing knowledge. So that I adapted that out of AOP, in particular for this purpose here of the DILI. And normally it starts with a molecular initiation event where a compound with a particular physical chemical property that's taken up, absorbed, distributed, excreted. It has some metabolism. It's then exposed to the cells, to the subcellular fractions, to the mitochondria, to the endoplasmatic reticulum, and there it causes some specific mechanistic effects from covalent binding, and then that can be then the cause for the immune stimulation. At the level of the organ, as I said, you can have all these histopathological changes, which are important and first diagnosed in animals under normal conditions, but also at the level of the whole organism, we can look at adverse outcome pathways, since we know that that a patient, for instance, is responding in a different way as a healthy subject or even as a healthy animal. So, and these aspects, they can be addressed at the level of the individual patient, but also of a whole population, where we know that we have here some genetic, epigenetic disease, underlying diseases, and other factors playing a big role here. So these are the building blocks for the 3D models, which can be investigated for the cellular key events for the DILI. So these are the building blocks for the three major pathways of toxicity. So the first one is the mitochondrial toxicity and the rust formation. This is described in the literature, and we have here a sequence of events which we can follow in order to understand the mechanism of DILI much better. The second one is based on the disturbed bile acid homeostasis, where we have here some efflux inhibition, and the intracellular bile acid is then inducing here some cholestatic and specific conditions for cytotoxic events. The third one is the haptin formation, which is a result of reactive metabolite formation, the covalent binding and adduct formation to proteins, which then are taken up and presented by antigen presenting cells, and then activating T cells, causing inflammation, and then also killing the cells. So these are the building blocks. And how do we come to this sequence of events? So one simple way to do that is just looking on different biomarkers and evaluating at what time point these biomarkers come up. For instance, here where we see the green arrow is, you see that, for instance, reactive oxygen formation can happen within a few minutes, in fact, yeah. And the result of that could then stimulate, for instance, apoptosis after 3 hours, and that could induce then the necrotic events as a secondary event. So these biomarkers, the sequence of events, gives already some key hypotheses which are important, but they do not explain exactly what really happened under these DILI conditions. So, which means we have to add here another layer of evidence for the validity of the mechanism, which is the causality assay, and these are again the biomarker which gives the sequence of events. And for the causality assay, we need to be very pathway specific, and we need to dig out this information by the so-called causality testing or causality assay strategy. This is nothing else than working with agonist or antagonist, is what you normally do in clinical in pharmacology, classical pharmacology, and also toxicology, that you're using an antagonist or an agonist, which is a specific for a pathway, and that you're looking then on a specific, focusing that on a specific mechanism. And then in this case, what I'm showing you here is that we're using the readout of the ATP LDH assay to test specific pathway antagonists and agonists, and that will then indicate the validity of this pathway for the overall DILI mechanism. So these are the again the building blocks together with the possible causality assay which we will have, and I will give you now an example of those assays. And I will start here first with the the reactive metabolite oxygen rust causality assay, where we're using as a modulator of the endogenous situation, the pathway, a modulator of the glutathione synthesis, in order to get some information on the formation of reactive metabolite formation or rust formation. So by depleting the glutathione, we could potentially modulate the the effect of reactive metabolite. Once the glutathione, which is at high concentrations in the liver, and it has its function is to detoxify electrophile metabolites, and then keeping the liver clean. And as long as we have a high glutathione level, the liver is normally is nicely protected. But when we're starting to deplete that, we make it more susceptible for RUS attacks as well as for reactive metabolite attack. For this causality assay testing, it's important that we do not kill the cells itself by the depleting glutathione compound, in this case the butyrylene sulfoxamine, which is an inhibitor of glutathione. And you see on the right panel that that after plus minus BSO is the same, but when you for the ATP, but when you look on the level of the glutathione, under these conditions the glutathione is completely depleted. Now we're using here a typical example of a compound which is the best, let me say, or one of the best investigated compounds, the acetaminophen. It has a CYP23A, 2E1 mediated reactive metabolite formation. When we tested under glutathione depleted conditions, then the cytotoxicity curve gets a left shift when we're looking on the ATP cellular contact. When we're looking on the right side on the leakage of the cytoplasmic LDH, then we see that the BSO condition is enhancing the effect by producing more membrane damage. So this shows that we have here a positive confirmation of our hypothesis. We have reactive metabolite formation and also reactive RUS formation, which could play a role here in the pathomechanism. So let's look here on the next causality assay, where we have also reactive metabolism. In this case, it's the aflatoxin B1, which is producing an epoxide. And it's known that this epoxide comes via the 3A4 metabolism, and that this is causing the carcinogenic effect of the aflatoxin. But it's also known that this reactive metabolism can then also be toxic. And we investigate by investigating the cytotoxicity under two conditions: with an PAN inhibitor, the 1-ABT, and without the PAN inhibitor, we could demonstrate that we distinguish the cytotoxic effect. So there's a steep IC50 curve, which is 0.2 for without the ABT, but with the ABT we cannot even measure it. So there's a huge shift of the IC50 curves. That suggests that we have here some reactive epoxide formation. Now I'm coming now to another causality assay. It's a bile acid causality assay, and here we investigating the effect of the bosentan, which is an endothelin antagonist, antagonist, and we're looking here on the secretion of endogenous produced bile acid. So normally you can also load up cells with artificial bile acids, and then you load them up, and then you measure the secretion afterwards, so after the treatment with the compound. So this is something we have also tried, and there are several substrates which are possible to use here. But it turned out that these type of assays, they have a high variability, and they do not give that robustness as we expect that from our spheroids. So then we were thinking about how could we improve that that important information then also for the testing of the DILI, and we came to the proposal, or we investigated endogenous bile acids which are produced in the spheroids themselves. You have here some bile acid synthesis, and we looked on the effect of the inhibition of bosentan on the secretion of these endogenous bile acids. So it's an important control experiment that you also look on the cytotoxicity. Once you're killing the cells, you would certainly then also see a decrease then in the secretion But in this case, we were out of the cytotoxic range, and you see that we have here a very specific response of inhibition of bile acids, the GCA, as well as on the GCDCA. So that gives some confidence that we are looking here on the right thing, and a negative control, which was also DILI negative, and this was also an endothelin A inhibitor. It's the ambrisentan, and that had no effect. So data I have not shown, but I can tell you that it is not positively affecting the secretion of these bile acids. So now there's another way to look at it, by doing a bile acid causality assay, where we're adding some bile acids in this assay, and the composition of the bile acid is exactly what you can measure also in human plasma. When you add that at non-cytotoxic concentration, you see here that in combination with the bosentan, this is enhancing then the bosentan effect. So that means we may have a situation where we have the endogenous production of the bile acids. They're accumulating intracellularly, and the addition of the bile acids on top of that, causing a situation which is a disease-like situation, where we then getting the endogenous cytotoxicity of the bile acids, and that's confirming that we have here, or indicating in the direction that we have here some bile acid type of mechanistic DILI formation. So I would like to come to the next causality assay, which is an inflammation causality assay, where we're adding LPS together with the trovafloxacin. And maybe all of you know the classical experiments. I think were first done with Bob Ross. They tested the LPS and mice with the trovafloxacin, and he could demonstrate that the liver toxicity was enhanced by the LPS. We could recapitulate that this experiment under our co-culture condition, where we have hepatocytes and Kupffer cells, and we're stimulating here with the LPS the Kupffer cells, which is then enhancing the cytotoxicity of the trovafloxacin. So when we do the same experiment with the monoculture without the Kupffer cell and adding the LPS on top of that, you see on the right upper panel that there is no difference as we observe that on the left side. So you can also do that at the level of the LDH, not at the cellular content, but at the level of the extracellular component LDH. You see that this effect is enhanced in the co-culture, but not in the monoculture. So that shows that we need the LPS. That shows that we also need the Kupffer cell in order to dig these effects out. We can do that also with a negative, in vivo negative compound, which is structurally related. It's a levofloxacin, and you don't see that the LPS enhancing the effect in none of the models. So that gives also some confidence that we can exactly in the same direction, we can mimic the in vivo experiments as we can mimic the in vivo experiments by the in vitro experiments in the 3D culture conditions. So difficult. In order to be more mechanistically specific, I added a slide for the inflammatory markers, which we can measure on top of that. We have not done that in this experiment, but we have seen it also in some customer studies that it is also increasing. We can show that here in our NASH model, where we compare the NASH conditions versus the lean conditions. Also in the NASH model, we have these inflammatory components, and we see that we are very specific in terms of the secreted cytokines and chemokines, and that we can do that also at a level which is highly scalable and which is highly reproducible, and where you can also get some highly statistically significant differences in terms of the outcome. That's where we are. I have here also another case, which is the so-called steatosis causality assay. In this assay, we are inducing steatosis, which is relatively difficult to do by some factors, dietary factors. Some compounds are also inducing steatosis, but the purpose here was just to induce, independent from a compound, steatotic conditions and then testing then the effect of a cholestatic compounds. Then on top of that, and we were using here the chlorpromazine, and you see that the cytotoxicity under this steatotic conditions is strongly enhanced, and in the concentration range where you would not see something with the chlorpromazine itself. So the steatotic conditions were also confirmed at the different level, at the high content imaging level, and quantitatively at the level of the triglycerides. So that shows that we have steatosis. Steatosis is a risk factor for DILI, and this is an opportunity now also to explore that more systematically, and if this is more fundamentally, let me say, confirmed by testing of more compounds, such an assay could help you also to stratify patients, where we have here a patient situation where we expect here some interaction with some potential cholestatic compounds. So that's a possibility. I'm coming now to another causality assay, which is very important, since it's also bringing the immunology, the immune properties on a chip, which is one of the latest, let me say, achievement of the 3D culture, where we're bringing here some PBMCs together, which contain monocytes, lymphocytes, T lymphocytes, killer cells, and other type of cells, immune cells on the dish, together with the 3D microtissue. And I think it's a nice combination, since we using the microtissue, the liver cells, to produce some potential reactive intermediates, which could covalently bind then to proteins. The proteins, they are taken up by antigen presenting cells, and then they are presented to T cells, and they're activated then further, and then this is killing then the cells. So this is a new type of assay, which is very important, not only for the DILI itself, but also which is opening a strong perspective for the testing also of biologics, which cannot be tested neither in animals, or some of the biologics cannot be tested, and here we have the possibility that we also extend the range of applications, from moving from small molecules also up to biologics. So I would like to show you some examples. So this approach is investigated not only in the liver, it's applied in the pancreatic islets, and in immune oncology it's also used. And you see in the upper panel the time dependency from T0 to 3 hours and 8 hours. And you see that the microtissue, in this case, it's a pancreatic islet. There happens nothing over this period of time. When you're adding then the PBMCs together to the spheroids, you see not happen much. You see the PBMCs, they're aggregating slightly, but when you're activating the PBMCs with the CD3 and 28, then you're seeing that the T cells are activated, and that they're slightly starting to invade into the spheroid and starting to destroy it. So that's in fact the principle of this assay. In a nutshell, so the human microtissue, the causality assays, they can be adapted to a wide range of pathways to explore specific pathway information by using specific pathway modulators. So that gives us specific mechanistic information, but it allows us to take out one of these mechanisms and then use it also for mechanism-based screening. I would like now to come to the impact to another dimension of those 3D microtissue assays. So we identified clearly the toxicology as one of the major applications. Then we also have to consider the clinical situation, and also regulatory is playing a role here. For the toxicology, I think the value is clearly in the early phases with hazard identification. We'll be using front-loaded assay. For the later development phases, once we have in vivo data, the mode of action is important. The question is, how is the translation to man? Clinical biomarker situation plays a role, and also mitigation strategy can be initiated already at that point. And also for the backup support, this is important, besides the importance for the 3R. So now I would like to shortly talk about the translation to man. And this is what toxicology does, and that's what we are doing to bring a compound into the clinics to IND and first in human studies, that we're looking on the therapeutic window, the safety margin. So we're making the concentrations and the exposure of the animals and calculate that to the human. And once we have a sufficient therapeutic window, then I think it's a clear go. There will be no one against it. But if we have something which is very close or even lower, then of course some traditional companies they stop completely. Or in the meantime, if we have an interesting profile of a compound, a new therapeutic principle, a new mode of action, then the question comes up, shouldn't we go further? How can we do that? So, and I think here cross-species comparisons play a role here, which is starting by the recapitulation of the in vivo effects by in vitro studies. In this case, this is an example done for customers where we had an effect in the dog. It was central lobular necrosis and increase of ALT, and we were recapitulating that at the in vitro level, and in the same way. And the question was, how did it confirm then to the human situation? In this case, it was a clear it was negative, and the compound had the same response under these preclinical conditions, and it was negative in the human, and that shows that the effect was not translatable from the dog to the human, and that gives a clear go situation for this compound. So I show you here a case number 2, where we had another effect in the dog, where it was a microvesical steatosis, and we could confirm that then also with this compound B, and there was no difference. But in this case, we had here an effect in human, which makes the compound B not so interesting, and since there was this uncertainty, it led to a no-go decision, but it didn't lead to the situation that the chemist was not interested anymore. Particularly these cases, there are interesting cases where the chemist really wants to recapitulate the effects and wants to modulate the molecule and bring it and to improve it. So here's an example of such a cross-species comparison. That's also an endothelin antagonist, and it was taken from the market, and it was not possible to make here a good selection for based on the preclinical outcome, and which was in fact pretty much clean. But under clinical situation, it turned out humans are having an issue with this compound causing DILI. And when you look here on the red, it was really flat. There's no effect. In the monkey, it was also really flat, except the high concentration. In the dog, you see that there is a slight effect, but it's clearly stronger under the human situation. So that shows the higher sensitivity of the human model. The difference between rat and dog and human can also be shown on the level of the species comparison for the aflatoxin B1, and that was a case in the early 70s and 90s in India and in the US, where animals died, dogs died because of polluted food, and this is recapitulating the stronger effect than in the dog. So I'm coming now to the clinical situation. Be comparing healthy animals to diseased patients, and I think we need to correct the safety margin for the diseased patients, because we know there are some factors which are changing the effects of a drug, which can be age, can be the BMI, can be the gender, and it can be the underlying disease. So that we have certain risk factors which enhancing the effects, and we make also the unpredictable event likely to occur. So how can we investigate that? So the answer is we can investigate that by customized microtissue production, that we're taking the microtissues from the diseased patients and compare that to healthy patients. We can do that for particular population, that we're looking at ethnicity, that we're looking on polymorphisms, that we're considering responders and non-responders. And if we have some genome-wide association study, we can address that. We can do that at the level of the individualized patient by using IPS-derived microtissues. We can do that at the level of the idiosyncratic DILI, as described before, with the PBMCs and with HLA-matched microtissues. We can do that by the PBMCs coming from the same donor. We can combine also the PBMCs with the DILI components, then also with the liver components of. We can compare the PBMCs from the DILI responders with the microtissue. So that gives us a good insight into the underlying mechanism. So I don't want to go into the mitigation strategy, which can be from the de-risking of high-low cases to the discovery of new biomarkers and to the antidote development. So one important aspect of all the work we're doing in toxicology is to give that basic understanding and making a better risk assessment, then also to our health authorities, which finally approving our data and helping here for submission. Now I'm coming to one of my last chapter, which is the current testing paradigm. So that's what we're doing. We're translating from animals to patient, knowing that we have patient is not one population itself. And I think we're learning by the biomarkers from the different patients on biomarkers to investigate that also under animals. But once we are getting to new therapeutic modalities, biologics, and we are looking on gene therapy, and I think we cannot do that in a way as we have done that over the last decades. Yeah, I think we need now to learn from the patient and address that more specifically to their need. And the patient tissue here helps to be more patient specific, more patient centric, and also taking all these aspects coming from the patient into consideration for testing new modalities. These are the challenges I see. It's to get into the field of the new therapeutic modalities, and I think with the PBMC model co-culture to the microtissue, we have here very good a good start. There's also some brilliant start for predictive toxicology when we combining the spheroids for their long-term properties also with the big data and using the artificial intelligence and deep machine learning to create here some new algorithms. Then in terms of single cell genomics, that's the next dimension where we could get more mechanistic information. And of course, for the gene editing part, I think we can using here more specific methods, we learning, and we can apply tools then which can be used then from target validation also into target validation. The microbiome is a highly important scientific field, but it's currently difficult to get here and hands-on, because you cannot combine microbes with mammalian cells, so that's not possible. And then we have certainly some huge expectation and also some possibility in the organ on a chip system, where we combining then cells from different organs, and to see how the interactions are. In particular here, there's a huge potential, because the systems, they are so easy, amenable. We do not need that much. We can do it at a high scalable level, and then that will then give a whole bunch of data. So in summarizing, so we have here a very pragmatic view to look here on drug discovery and how to use these model systems to make the translation and building the bridge to not only humans but also to patients. We can address a whole bunch of questions which can be applied flexibly to the needs of drug development. So the systems have a high throughput, they have high quality, robustness, and scalability, and I think this is an important aspect that we have here, microtissue technology, which is also prepared for the next generation of new therapeutic modalities. Having said that, I would like to stop here, thanking my colleagues for their contribution. Thank you very much.